So it's my pleasure to introduce Sergey Nizkorodov, who is visiting us today. He's from the University of California, Irvine, where Jim is going to go soon. He got his degree, PhD degree in chemical physics from the Basel University in Switzerland in 1997. And after that, for 3 or 4 years, he did a postdoc at CU in Boulder and then at Caltech in California. And he joined the faculty position in the Department of Chemistry at the University of California, Irvine in 2002. So his expertise is mainly in molecular spectroscopy, high-resolution mass spectrometry, and photochemistry. And so today he's going to tell us about his work on photochemical aging of organic aerosols. And I will let him. Podium?
Well, thank you very much for the invitation. So I would like to start by thanking my group members and also acknowledging my collaborators. We've collaborated with Alex Laskin and Julia Laskin from Pacific Northwest National Lab for a long time, and that's a postdoc Pen Ling. So you will see some data. And we are funded by NSF primarily, and they are funded by DOE. So I would like to start with what I view as interesting milestones in atmospheric chemistry. So Da Vinci already thought about atmosphere, so he attributed the color of the sky to scattering by particles and droplets, and then oxygen, nitrogen, major things were discovered, hydrogen. 1800s, argon, helium, methane, other trace gases were discovered. Then Eichen postulated that particles are required for cloud formation. And then, there was, ozone layer chemistry. So this is from Sir Sidney Chapman predicting the existence of ozone in the stratosphere and actually being accurate within a factor of 2 about it. So in 1950s, there was lots of interest in photochemical smoke, starting from experiments by Hagen Schmidt, the botanist who's done smoke chamber experiments. He put people in the chamber, plants in the chamber. So that a lot of fun stuff. So that's the equation that came out of that work. So then catalytic ozone destruction took off in 1970s, and so this is what actually UCI is famous for. So Roland, Ciceron, and people like that. So heterogeneous chemistry, the importance of it was realized in 1980s perhaps, and the key reaction, for example, happening in polar stratospheric clouds is the one that converts HCl and ClO and O2 into Cl2 that photolyzes easily. And so recently people have been focusing, so now we're in the aerosol phase. There is quite a bit of emphasis placed on aerosols, maybe too much emphasis. We'll see. But hopefully this problem will be solved and we'll move on to something even more interesting. So why aerosols? There are a couple of reasons. For a while, so people didn't believe they could be important too much in chemistry because of the very small weight percent. Even in a polluted atmosphere, that's what you're dealing with. There is not much that's floating around, but they have a very disproportionate effect on the environment. So they affect chemistry. So for example, reactive uptake of N2O5 happens on aerosols. They affect climate directly and indirectly. They affect health. And this is actually the least understood aspect of aerosol research. So why are they unhealthy to breathe? So this is the primary motivations for studying them. So we look mostly at secondary organic aerosols, and secondary doesn't mean secondary in terms of importance. It's secondary in terms of formation, and the way you make them, you start with a source that could be, let's say, a forest or an industrial source that emits a VOC, volatile organic compound. There is a classic route, so you oxidize it and then it condenses into particles. And there is another route, so you can oxidize it, that goes into aqueous phase, goes through aqueous oxidations, and water evaporates. You end up with SOA that people call aqueous SOA. So that's, the sort of thing that Barbara Chirp and Barbara Irvine are doing. And so what we do in our research, so these are the 3 driving questions that we are trying to address. So I'll deal with the first 2, putting more emphasis on the second. So the first one is essentially what's in this secondary organic aerosols. So they are complicated, and I'm going to show you how you can use tools of high-resolution mass spectrometry to tell you something about their composition. And the second one, perhaps more interesting, how do aerosols change in time, how they age. So everything ages, so including this podium, which will in 100 years fall apart. So the aerosols age on a faster time scale. What happens to them chemically, and that's what we are trying to understand. So the approach we take, so we have a small chamber. It's not as impressive as the one I've just seen. So it's a 5 cubic meter bag. There are some lamps, enclosure, some instruments. So we start with the VOC. We produce secondary organic aerosols. So that part is reasonably well understood. It's been done many, many times by many, many people. So once we make aerosol, it becomes our sample. So what we do, so we collect it and we study it, so using mass spectrometry, some X-ray methods, some normal classical analytical methods, and I'll focus just on mass spectrometry. And our mass spectrometry tool that we use, so this is where collaboration with Julia Laskin is a mass spectrometrist and Alex Laskin is a field scientist and for the atmospheric in the atmosphere. So they have an orbit trap based mass analyzer, so there is a C trap that injects ions and a linear trap to accumulate them, an electrospray source. So I'm going to in reverse. So electrospray is a soft ionization technique that either deprotonates your molecules or puts a sodium or potassium or proton on them. And then in the linear ion trap you can do tandem mass spectrometry, and then orbit trap is a very accurate analyzer. So you measure, the accuracy is about 1,000th of a Dalton, so resolving power is 100,000, and it turns out that that's enough to resolve quite a few molecules that way. So I'll give you some examples, applications of isoprene. So I had a very talented student coming from my group, Tren Nguyen, and she later went to Caltech and will start her own faculty position at Riverside this summer. So she published a bunch on that molecule, isoprene. And isoprene is important. It's quite important as an SOA precursor. And so what we wanted to know is the molecular level analysis of isoprene aerosol and effect of environmental conditions such as humidity on the composition. Okay, and so here is an example illustrating what high-resolution mass spectrometry can get for you. So you make an isoprene here from using ozone as an oxidant. Not the most common oxidant for isoprene. It's just an example, though. You get a mass spectrum. It's an electrospray ionization mass spectrum. You zoom in around a peak, let's say one of them, 250, and you actually discover that it's not one peak, but several clustered at around the same mass. And the peaks are narrow enough that you can unambiguously say that this is, for example, C10, H12, or 6, and that's so you resolve all of these compounds. So if you use, for example, time of flight analyzer of the pretty decent resolving power of 5,000, you would see that. So you would not be able to resolve any of that. So you end up with a giant list of formulas, and it can be overwhelming. But we've learned to look at those lists and look for patterns in them and see how those patterns change in response to conditions. And that's how we do most of our experiments. So here is an example of this pattern recognition. So for example, in isoprene plus ozone secondary organic aerosol, you can replot the molecules. You can see in what's called a Kendrick diagram. So there is a mass of the compound, and that's a mass defect on this axis. So the mass defect essentially shows you, so if you have a constant mass defect, you have the same family of compounds. Okay, and we've tried different, and you can do this game with different bases. So we've done formaldehyde here. It turns out to be the best base for isoprene aerosol, and we actually give it a chemical interpretation that a lot of these products are actually accretion products of formaldehyde reacting with either alcohols. So that's a major channel. So formaldehyde reacting with alcohols and these sort of compounds and hemiacetal stay inside the aerosol. So we see lots of them in there. So here we look at the effect of NOx. So we make isoprene from OH, isoprene aerosol from OH reaction with NOx and without NOx. So without would be pristine conditions. With would be, let's say, conditions affected by a city. Here is two mass spectra. That's how they compare. I painted nitrogen-containing species here in red, and there is actually quite a few of them. So if you count all of them under NOx conditions, this is number of nitrogen atoms, 0, 1, 2, 3, 4. So So the majority is still nitrogen-free, but under high NOx conditions, you get maybe up to a third containing nitrogen. Under low NOx conditions, you still see nitrogen because it's impossible to get rid of nitrogen in a chamber. There is always outgassing from the walls. And it's interesting, actually, if you look at the species, you see, using this Van Krevlin diagram, so that shows oxygen to carbon ratio in a molecule, hydrogen to carbon ratio in a molecule, and each point here is a molecule. And so the red ones are the ones you make under high NOx conditions, the blue ones under low NOx conditions, and the black ones you make them under both conditions. The point here is you end up with a completely different subset of chemical compounds in your aerosol. So it's quite interesting. So this is the one I didn't expect. So there's a Tran said she wants to look at the effect of humidity, and I told her not to waste your time because people have tried it and found nothing interesting, but she did it anyway. And indeed, so if you look at the SOA yield, so you start the reaction, one of them is humid, one of them is dry, and there is essentially no difference. You look at the development of major species, formaldehyde, acetone, acetaldehyde, I believe. I don't remember what that is. There is really no difference between humid and dry conditions, but then you look at the particles composition, and the difference is quite drastic. Okay, so for example, organic nitrates were suppressed by 40% under humid conditions, possibly due to hydrolysis, and oligomers changed quite a bit. Sorry, this is a complicated figure, but this is an extent of oligomerization in the product. So, for example, here we are looking at a family where you start with this compound and you add units of 2-methyl glyceric acid, which polymerizes it's a major building block for isoprene aerosol, and you can add 8 of them. So the black ones is what you get under dry conditions, and white ones under humid conditions. You really suppress this oligomerization under humid conditions. Even though the yield stays about the same for aerosol, the types of compounds that sit in it are completely different. It's very interesting, and this is the actual reaction. So this is this one is a condensation into esters. This is the one that's affected by humidity because water is a product, and that formation of hemiacetal doesn't care because in a hemiacetal reaction you don't produce any water, so you don't shift this equilibrium by humidity. Okay, there is a lot more, but I promise Alma to focus on photochemistry. So, and so the next question I want to discuss is how the composition and physical state change with time, so the aging. And the one type of aging that we look at the most is photochemical aging. So essentially, you start with a particle or maybe a cloud droplet, fog droplet, could have complicated composition. You expose it to solar radiation, and you have condensed phase photochemistry happening inside the particle. Okay, so we want to know how fast it is, what happens inside, and so what sort of products. Does it have any effects on the, let's say, size of the particle and phase state, chemical composition, optical properties, toxicity, taste, smell, you name it. So well, we don't do the taste bit. It's kind of dangerous. So what's the current state of knowledge? There is not much. So I participated in writing this review. So I was responsible for writing the organic photochemistry bit for this review. So it focuses on heterogeneous photochemistry in the atmosphere. And essentially what we stated there is that there is a huge body of organic photochemistry literature. Huge. There are books, big books. Very interesting, actually. They provide fundamental understanding of mechanisms, but it's all 254 nanometer photochemistry. Okay, mercury lamp emission, so which is relevant for the upper atmosphere, not so relevant for the lower atmosphere. So also they use bizarre solvents for the photochemical. Their goal is to optimize the yield of a particular photochemical reaction. Our goal is to study the dirt that is produced as a byproduct, which is, a secondary organic aerosol is a dirt that's a byproduct of major reactions, essentially, right? So we have different goals. So there is a huge body of dissolved organic matter and aqueous photochemistry literature, but it's very useful again for clouds and fog processes, but it turns out I'm learning that dissolved organic matter and secondary organic aerosols are quite different in properties. These tend to be much bigger molecules than those, and they actually lots of supramolecular complexes in here, and not so much here. Also, aqueous phase and organic phase in SOA mechanisms are quite different. Finally, there is some organic aerosol literature, but very limited experiments, and to the best of my knowledge, there is really no serious models that include any sort of organic photochemistry into their models, and I hope you would do that, or already did. So I'll start with aqueous secondary organic aerosols. So we oxidize it, VOC, so that it goes into aqueous phase. There is a normal OH in the aqueous phase and ion and phantom chemistry. But in addition, we'll also look at the direct photolysis. Very straightforward experiments to do. So you can have both direct photolysis and OH chemistry. Those are hard to separate. We try to separate them by doing low hydrogen peroxide. Well, we don't add any OH precursors into the solution, or sometimes we do and then compare. And again, so we use a chamber, we make aerosols. This time we extract it in water and photolyze it in this window. We do very simple, let's say there is a cuvette and a UV spectrometer. Photolysis light comes from the top. There is a reference. We track absorbance, and then we inject the solution into this Orbitrap mass spectrometer. And so in our first papers on this, so this is the first paper we published on this topic. So Adam, so again, talented graduate student, postdoc at Harvard right now. So he made aerosol from limonene and ozone reaction. And in high-resolution mass spectrometry, you can go through every structure formula that you see and assign the average formula to the whole aerosol. So that's the average formula of the aerosol. So if you, after interaction with radiation, that's about 24 hours equivalent of radiation. So what you end up doing, you end up destroying bigger molecules, creating smaller ones. Okay, so the oligomeric compounds tend to be destroyed, and then Tran followed up with isoprene aerosol work. So the isoprene, she made it under high NOx conditions, so it had some nitrogen in it. So she irradiated it, and the most interesting finding of that work was actually the nitrogen compounds were quite sturdy. So either the nitrogen was coming into the molecule, so they were just not removed very efficiently. So we had quite a build-up of nitrogen compounds at the end. And some of them had really bizarre structures. So I've never seen, we've analyzed this. They had, I'm not going to show any formulas, but they're scary organic molecules that you produce in the process. So more recently, so Diane Romanowski, so she's an NSF fellow, also very talented student. So she said, okay, let's do a survey. So I'm going to take all of these molecules, I'm going to photooxidize them by OH under high NOx conditions, or ozone, or just OH, and then put them in water, photolyze them. For each one, do high-resolution mass spectrometry, and do, look at the effect of photolysis on the composition. So this is how the high-resolution mass spectra of all these molecules look like. If you like looking at mass spectra, here they are. Some people really like them. But what you learn from them, composition is complex. Between all of these mass spectra, there are many common peaks. So really, ozone chemistry is very destructive. So you lose memory of what you studied with very quickly. And the major products that we see is that's very important. So the hemiacetal coupling products, there are esters, and there is some evidence for Kriegi. That's probably an artifact of the chamber, unlikely to happen in the atmosphere. But on the higher concentration of the chamber, there is quite a bit of those products. And this is high NOx, OH oxidation of the same things. The orange ones contain nitrogen. So here it's a bit more gentle. The composition is still complex, but the products are unique. Actually, by looking at the mass spectra, you can tell what you started from. And we need to develop that further, because no one really uses high-resolution mass spectrometry to do any sort of apportionment of aerosols or splitting. So there is oligomers, there is nitrogen-containing compounds. But this is what the photolysis does. So let's take aqueous alpha-pinene SOA. This is the mass spectrum, molecular weight. I believe before, the green one is after, and the red one is the difference. And the fact that it's not huge, but that's only 1 hour of irradiation. Okay, so and what you tend to do, you tend to destroy higher molecules and build up smaller molecules. Again, we see this pattern in which our photolysis is destructive towards oligomers in the sample. So it's interesting. So there is a big community of people using what's called an aerosol mass spectrometer, and what they can do, they can measure average properties like average oxygen to carbon ratio, average hydrogen to carbon ratio in the sample. Those don't change at all. So you have quite a bit going on the molecular level, but on average, your aerosol composition doesn't change. That's another way of looking at the same. This is the type of aerosol that we started with. And this is the average carbon size, let's say for alpha-pinene oxidized by under high NOx condition. You start from maybe 17 carbons, you end up with maybe that many. So here is also destruction. This is an interesting case. Glycol didn't care about radiation. Humulin didn't care. 3-methylbenzene didn't care. So in average, we also find this aerosols produced from aromatic compounds. Even though this is the most absorbing aerosol and that's the next most absorbing aerosol, they don't respond to photochemistry. And I'll try to explain why later. And we generate tables of data like that, and you don't need, there will be no quiz at the end or anything like this. You are not, it's just to show you that, this from high resolution mass spectrometry, you could get all of these formulas. But the big conclusion here is the molecules get reshuffled by radiation quite a bit, but the average level of oxidation doesn't really change much as a result. Okay, so we decided to be a bit more quantitative about this work. And we did a survey study and submitted a paper recently on that. And this was done by these 2 undergraduate students, Nujat and Maria, and Diane wrote the paper for them. The undergraduate students are great about doing experiments, but when it comes to papers, so please don't make us do it. Yeah, so the question was, what are the effective absorption cross-sections by these different types of SOA, and what are the typical photochemical rates? And they patiently took all of these precursors, isoprene, alpha-pinene, all kinds. They oxidize them by ozone OH, high NOx, so this oxidation. For each one, they collected it, they weighed how much they collected, and they extracted it in methanol. So we wanted to do water, but water does not dissolve many of these things. So we ended up using methanol and ended up taking absorption spectrum, repeating it many times, and then ending up. So you end up with what's called bulk mass absorption coefficient, which it's a measure of absorption coefficient for mixtures, which you don't know what are made for. So this is an example. This is glycol OH. So the values are actually quite high. So, but for alpha-pinene OH, so this is an order of magnitude smaller. Limonene ozone also an order of magnitude. So these are very small. So for example, they will not have any sort of direct effect on climate. So this one is high enough to be considered a brown carbon sort of species, but still on a smaller end. So then we take these mass absorption coefficients and we convert them into effective absorption cross-sections. What you need for this conversion is some sort of an effective molecular weight. Implicit in this, we assume that SOA consists of the same molecules with the same photochemistry, so they behave in some way on average with this average molecular weight. So you get some sort of a number for effective absorption cross-sections, but that's not all that you need. You need also quantum melts, and of course we have no idea what those are, and so you know well about this. So you have to improvise, and you can take limits. For example, we've taken hydrogen peroxide as one limit, quantum melt of 1, but then we cut it off at 400. And the other limit we've taken is acetone. So here is one atmosphere acetone absorption cross-section. And then we convolute it with, I haven't shown the solar flux, but it starts here and goes up. And then here is an example of absorption cross-section for one of those. So we convolute it and we calculate J photolysis rate as a function of solar-zenith angle, and then we average it over 24 hours. And so this is what you get for Los Angeles, since we live there. This is using hydrogen peroxide. This is as high as it gets because we use hydrogen peroxide quantum melts, which is 1. It cannot get high. This is an upper limit. And these are high, so you can see that the photolysis rate, well, those who are familiar, this is of the order of what NO2 does in the atmosphere, so pretty high. Even that is quite high. And so if you use more realistic, and then you calculate the effective average lifetime. So if we use more realistic ones for acetone, so we get 1 hour to several days, and qualitatively, so basically I talked to Sasha at the AGU meeting. So I think what you do, you take a whole series of quantum melts, and you have a nice plot how this is going to change as a function of the quantum melt you assume, and you arrive at a conclusion that must be important. Essentially, the same thing. All right. So, and this is the data that the cross sections we will just send to anyone who wants them. I think they're quite reliable. Okay. So let's now talk about organic photochemistry. So in this case, we oxidize organics, so there is gas phase chemistry going on, then it condenses, and then you have aging inside, heterogeneous multi-phase aging by OH, ozone, but also photochemistry. This is the part that I think people neglect, and they should pay more attention to it. And so we try to do experiments to figure out how important that bit is. And here, it's a challenge to do it in a chamber, for example. If you do it in a chamber, you have an aerosol that contains particles and vapor, and guess what? That vapor is dominant. Okay, so the mass concentration of particles is minuscule compared to mass concentration of the vapor. So you radiate the whole thing. How do you know what you're studying? Vapor photochemistry or particle photochemistry? So we try to get around that in several ways. So the one way is to collect secondary organic aerosol material on a substrate and anneal it to get rid of the vapors, and then study that material. That's what we do the most. Okay, so the disadvantages here, the advantage is no more vapors. The advantage is no more vapors. The disadvantage is that there might be transfer limitations. Maybe oxygen cannot get into the material, other things, and so on. But some people ask me about the substrate effect, so those don't matter. There is flow reactors, so you can strip gas. I'll show you some of that. You can strip off gases and just let particles go through flow reactor and do radiation there. And then you can do it in a batch reactor, but this is where you have to worry about these complications from VOCs. But these are the people who've achieved some useful conclusions using the batch reactors on aerosol photochemistry. So our early work, so this was one of my first graduate students, Maggie. So she's actually project manager at the National Academies of Sciences now. And what she was doing, she was making aerosols, collecting it on a substrate. So that's aerosol from limonene, irradiating it. And she was just looking at the emission of one thing, formic acid. So you irradiate it, formic acid starts coming out. It's a product of many reactions. And so what we've done, we've measured, I forgot what these points are. This is photodissociation spectrum. So you measure how much formic acid you produce at every irradiation wavelength. Okay? And the photodissociation spectrum actually matched the absorption spectrum, which meant that at any color of photon that's absorbed, you initiate chemistry that then results in formic acid. And of course, we've done control experiments where you just do visible or just heat it up, formic acid doesn't come out. It's really a photo product. But we have not quantified it in any sort of way. It's not just formic acid. That's another student I had, Steve Mang. So he collaborated with Don Blake group. The Don Blake's group specialty is VOC analysis by gas chromatography. So we collected whatever we produced as a result of photolysis of SOA and analyzed it. There is a whole bunch of things coming out. So the carbon dioxide, carbon monoxide, methane, ethane, other things. as photoproducts. And we looked at those. Actually, most of these photoproducts are consistent with photochemistry initiated by Norrish type photochemistry on carbonyls. So we had a break from that sort of work for a while, but now we are revisiting it. So I have a new student, and he just got an NSF graduate fellowship, so very exciting. So what he does, he makes SOA in a chamber, then collects it on an impactor. It's a Moody that's rotating and distributing aerosols on a window. Then he puts window in an oven, maybe 40 degrees, and runs dry air through it so that it anneals, so get rid of all the things that are volatile, and then irradiate it. So we now use LEDs as UV sources, handier than lamps, and they emit one wavelength, so easier to interpret. And then you detect VOCs. So initially he wanted to use cavity ring-down spectroscopy, but he ended up using proton transfer mass spectrometry, and that's how his data are looking like. This is now time. And these are concentrations. This is proton transfer reaction mass spectrometry. So you have your aerosol, this is your background, you turn on your lamps here. So there is a build-up going to steady state. So we've investigated what this time is from. The time is from molecules getting out of organic phase into the gas phase. This is just transport limitations. The more material you have piled up on the window, the slower that is. And that's also interesting because we want to collaborate with people who study diffusion in aerosols and see whether glassiness of aerosols affects that. You turn it off, then it goes back down. So it's, here he looks at formic acid, acetic acid, aldehyde, acetone. And he's done it for a whole bunch of aerosols. Okay, this is an aerosol type, and he's German, so he's using German color scheme here. He tells me it's German black has this color. So this is alpha-pinene, low NOx, high NOx. Let's see, what does it mean? These are the emissions of different things measured by proton transfer mass spectrometer on a relative scale. I think he calibrated some of them, but he still prefers relative scale. So the biggest surprise, all of them emit things to various extent. This was the biggest surprise for us with Viacol. He repeated this experiment many times. This aerosol is just dead. There is nothing coming out. You radiate it, and it's just, there is nothing. So we thought about this for a while. So what makes this aromatic aerosols different? You oxidize them, and there are ring opening products that we know would emit VOC upon irradiation, because that's the type of products you get from monoterpenes. But we believe that the majority of the products in aerosol still retain the aromatic rings. So they look like that, and what they tend to do, you excite them, they go to a triplet state through interstitium conversion, and then the triplet state form ring substitution products and not gas phase VOCs, or relax. Okay, so they relax quite efficiently. So that's our explanation. It's very hand-waving, and I'm a physical chemist. I know it's not very satisfying, but that's the best we can do under the circumstances. So we do other types of experiments. This time, so Hanna Lignell started this experiment. So she's a student. She was a student from Helsinki. So now she's an air quality management district. And so what you do here, so you collect secondary organic aerosol in a window. And the question is, so we are going to now put a probe molecule inside our secondary organic aerosol material, a probe molecule for the chemistry of which we understand. And we are going to see how the photochemistry of that probe molecule is affected by the solvent, and our solvent is secondary organic aerosol material. So it's waxy, gooey substance. So here is an example. So she collects secondary organic aerosol. This is this white blob of material. She then drops this is 2,4-DNP is 2,4-dinitrophenol. I'll show you the formula later. And you make a thin film out of it. Her second model system, so we collect secondary organic aerosol from limonene. So we brown it by reaction with ammonia. So I don't have time to talk about it, but these are nitrogen-containing compounds that you form. You make a film out of it. And then you irradiate it. So she puts it in a box like that. So here is her window. Here is radiation, and she takes the absorption spectrum. Very simple. So this is, we tend to do much simpler experiments than you do here. And so here is the scientific hypothesis. The hypothesis is that if the lower the temperature or lower the relative humidity, that film will become more viscous, and therefore maybe will slow down photochemistry inside the film by just keep constraining the molecules and not allowing them to move. So this is our first probe molecules. This is 2,4-dinitrophenol, and the way this molecule does photochemistry, you excite it, goes to triplet state, it looks around for a suitable OH donor, which it could be aldehyde or alcohol, grabs a hydrogen atom from it, forms this species. This is unstable and keeps producing products. So, and eventually this group becomes an NH2 group, aniline group. So this is how it happens experimentally. So you start with a transparent film of that molecule, and then you irradiate it, it becomes brownish. And in terms of optical spectra, here is the absorption peak of your 2,4-dinitrophenol. Here is this absorption spectrum of the product that comes up. So the product is an aniline product. And so for the second one, so we've studied this reaction quite a bit. So we make SOA from limonene and ozone, and then SOA reacts with ppb levels of ammonia and browns. We have no idea why what is in there that makes it brown. So we know only elements of the structures. We know the indole and pyrrole base, but that's it. So so the bones found it, and so this is the browning. You start with a transparent bit of aerosol, so now it's yucky brown, and then so Julie Lee found that at least in water, you put it in water, you irradiate it, that brown color disappears very quickly. So it's not very stable. So we wanted to look at it in a film. So in a film, so this is a characteristic absorption band associated with this brown carbon. So you irradiate it, it goes down, and so we also are able to measure the rate. So this is the measurements. What are the results? So what we do these measurements at different temperatures. This is an Arrhenius plot, and this is the rate of photodegradation, and the slope of this plot tells you about activation energy for this photochemical process. And so what's interesting, so let's start. Let's say this is the green one is an activation energy in octanol. Octanol is a viscous liquid, but flows. It's not too viscous. It's not too bad. And there is almost no activation energy, meaning it's really there is no intrinsic barrier to this reaction. But what's interesting, you put it inside the secondary organic aerosol, and the slope changes, even though the mechanism is the same. It's the same abstraction of hydrogens from alcohols in the matrix. But the activation energy becomes higher. And so our interpretation was that, we see this slowing down. So our interpretation was that we have increased viscosity that hinders molecular motion, and therefore that excited molecule can no longer meet a hydrogen atom donor. So, okay, if this works, so we should be able to, without changing any temperature, modify the rate using relative humidity. So here we keep the temperature fixed, but we change the relative humidity. As you increase the relative humidity, the matrix of the secondary organic aerosol softens because water is a good plasticizer. So the molecules are now able to move. And indeed, the rate goes up. And you may ask, maybe it's just water doing it. If I do this reaction in pure water, the quantum melt is suppressed by 4 orders of magnitude. So water is not participating in the reaction. It's just making the matrix softer, allowing the rate to go up. So it all makes sense. So we are quite excited about this, and we are investigating it further. And we collaborated with a group of Alan Bertram, and they use what's called a poke flow method. So they take secondary organic aerosol material, a blob of it, and they poke it, so it becomes a torus, and then it slowly reforms a sphere. And from the poke flow times, so the seconds, that's how much time it takes to reform a sphere, and the function of humidity. This is alpha-pinene, this is limonene. So we find that alpha-pinene is a bit more viscous than limonene, and this is consistent with our interpretation for photochemistry. So finally, this is the final bit of my presentation. We also try to do some photochemistry directly without depositing aerosols on substrates. So using a flow tube. And Scott Upstein, so he's an engineer from Carnegie Mellon, so was a postdoc in my group, and then 2 graduate students picked it up, Sandy and Amanda. So what you do here, you start with a smoke chamber, and then you let your aerosol go through a giant set of denuders. There's a charcoal field, and the idea is that they're going to strip off ozone and strip off all of the VOCs that could potentially be photoactive. Or you can just do bypass. And then you inject them in a flow cell. Actually, there are 2 flow cells. One of them is a quartz flow cell, and the other is dark. And they're at the same temperature, and that's critical. Okay, so because temperature messes up aerosols quite a bit. And then they come out, and we do PTMS. Well, don't worry about the alphabet soup, but what PTMS does is volatile organic compounds. This, I don't know what that does, but it gives you some information about O to C ratio. For us, it doesn't do much because we know it's organic. Okay, so it just tells us it's 100% organic. Great. So and then as MPS, and then we do filter NMR and UVVs, and then viscosity measurements. And so we've done only a couple of experiments with that. I'll only show data for the published one. And so we have some more data for diesel, which is not as interesting. But for alpha-pinene ozone SOA, so when you irradiate it, so this is the radiation time. This is an equivalent time of in actual days, including days and nights in the atmosphere. So what happens? Your particles shrink, and the shrinking is not large. Actually, we're not talking about big effects here. But they do shrink, and that may have some climate implications. So what's more interesting, on the same time scale, there is quite a bit of chemical change going on, and there is even more change on the molecular level. This is just measuring total peroxides in the particles, and you essentially, maybe with a half time of 5 days, just destroy half of them. So, and that has health implications. So that's quite interesting. Okay, so in summary, what I was trying to tell you is that photochemistry in organic aerosols and in aqueous phases is still fully explored. So we have some qualitative data, but nothing really quantitative, and it's not modeled very well. Existing data suggest that photolysis of secondary organic aerosol compounds is quite efficient in aging of secondary organic aerosols. So there are significant changes on the molecular level, but fairly small changes in the average SOA composition. So this is interesting. So the large absorption coefficient is not a prerequisite for efficient photochemistry. You really need to look at quantum fields, and we really don't have that information right now. And photochemistry may slow down inside viscous matrices, but again, so we don't know enough about this to tell how significant this effect is. But you could imagine a scenario, for example, you produce aerosol, and then it's lifted up in one of these big circulation events into the tropopause. Now it's cold, and things just lock in place because the particles are now cold and glassy, and now not enough photochemistry goes on. And I didn't talk about this. So we investigate brown carbon quite a bit. So essentially, brown carbon is poorly characterized organic molecules that absorb some near UV and visible radiation, and people believe they might contribute to direct forcing by aerosols. And you make it, for example, by burning stuff. So we published some papers that argue that it's not enough to just know absorption coefficients of brown carbon. It's also important to know how quickly it ages, and what photochemistry tends to do for brown carbon. It tends to photobleach them, okay, with different rates. So some brown carbon species are quite resilient to photochemistry, but others disappear in no time. So there are extreme examples where you can have brown carbon disappear in minutes upon irradiation. You form it during nighttime and completely destroy it in the morning. So very interesting. So I'll be happy to answer questions and show you any of the hidden slides I have. I have hundreds in there.
Time for questions. And you need to use the micro. It's recorded, so.
Yeah.
So, Jeff.
Hey, thanks, Sergey. It was a really fantastic talk. So, just thinking in general terms, and I won't hold you to your answer, how much would you say that oligomerization protects molecules from photolysis, or would you say it enhances it? 'Cause if you're taking like a carbonyl and a peroxide, making a peroxyhemiacetal, you're essentially removing functional groups that might absorb. So, is there an effect there?
Well, if I were, let's say, if you have a sort of constant functional group density per carbon, okay? Let's say you have a certain number of carbons in the molecules, but probability of having a carbonyl or peroxy group is fixed. Then a bigger molecule will have more functional groups in it, and just by having more functional groups, you will enhance the probability of direct photolysis effects, potentially. So on the other hand, the bigger the molecules are, the more viscous they are. So it could go, if viscosity slows down photochemistry, that can actually protect them. So I guess it's case by case dependent. I don't know if I'm answering your question.
Oligomerization specifically, I think in one of your slides you mentioned that oligomerization increases absorption. Isn't that right?
No, so I have not stated that. So, so I was talking about the effect of relative humidity on the extent of oligomerization in aerosol. So the dry relative humidity promotes more extensive oligomerization. And then photolysis tends to remove oligomers, so destroy them faster than smaller molecules. And that could actually be the reason why when you do measurements in the field, you see oligomers, but they're not as abundant as in lab experiments, for example.
Because the other thing I've heard is that by making oligomers, you decrease, you remove the chromophores, and therefore you take away any absorption or photolysis. But what you're saying seems to be the opposite, that with the oligomers, you have more absorption.
Well, it depends on what the chromophore is. Yes, let's say if your oligomerization happens by converting a carbonyl group into a hemiacetal group, yeah, you remove the carbonyl chromophore, but isolated carbonyls are lousy chromophores. Extinction coefficients are small, so they don't. On the other hand, in oligomerization reactions, you can make accidentally molecules that have absorption coefficients orders of magnitude higher than the molecules you started with. So we've seen it in some cases. So you build an array of products, and one of them is hugely absorbing and controls the absorption spectrum of the entire mixture. So that can happen. So I would not generalize that by making oligomers, you make the particle less absorbing. It depends on the chemistry.
More questions? Okay, so let's think one more time.